Hello, everyone, and welcome to GemPharmatech's webinar. My name is Komei, and I'll be your moderator today for today's session. We are delighted to have you join us today as we explore one of the most pressing challenges in neuroscience research in amyotrophic lateral sclerosis, or ALS. ALS remains one of the most difficult neurodegenerative diseases to study and treat, with limited therapeutic options currently limited to patients. To bridge the gap between basic research and clinical treatment, researchers require robust, reliable, and translatable animal models. Today's webinar will highlight how GemPharmatech's genetically engineered mouse models recapitulate key aspects of ALS pathology, empowering researchers to investigate disease mechanisms, evaluate novel therapeutics, and ultimately accelerate the development of effective treatments. And before we move on, I would like to quickly give a brief introduction to our company, GemPharmatech. GemPharmatech is a global provider of integrated preclinical services built on the world's largest library of genetically engineered mouse strains, with over 25,000 currently in our library, along with an advanced in vivo platform support. Supported by a team of 1,500 scientific experts, our infrastructure spans 7 subsidiaries across China and the US, with large-scale SPF bivariance capacity and over 6,000 new models generation capability annually. We are also able to conduct more than 1,000 preclinical studies a year, covering a range of disease indications that include neurodegenerative diseases, immuno-oncology, vaccine development, and more. Whether you need breeding, custom model design, or full in vivo services, we provide integrated, scalable support. Our newly expanded capabilities in the US position us to partner more directly with biopharma teams in the North America, navigating early discovery through IND-enabling work. And now it's my pleasure to introduce today's speaker. We are excited to have with us today Dr. Shan Lin, Study Director of the Neurological Pipeline at GemPharmatech. Have her walk us through the latest progress, insights, and applications from this groundbreaking project on ALS. Dr. Lin, the floor is yours.
Thanks for your introduction, and hello everyone. My name is Shan Lin, and I am the study director from Neurological Disease Pipeline in GemPharmatech. Very glad to be here to introduce the mouse models and the preclinical service platform for neurological disease research in GemPharmatech to you. Today, our topic is about the mouse models used in the preclinical research for amyotrophic lateral sclerosis. Before we begin, I'd like to briefly introduce the GemPharmatech's neurological platform to you first. To support the mechanism research and drug development for neurological disease, GemPharmatech has independently developed a series of mouse models related to this disease. Currently, our platform can be divided into the following main sections, including the neurodegenerative disease mouse models, pain disease mouse models, psychiatry disease mouse models, mainly in the COMS-induced depression models. And the last one is the neurological-related rare disease mouse models, including the amyotrophic lateral sclerosis disease mouse model and the Huntington's disease mouse models. For clinically common neurodegenerative disease like Alzheimer's disease and Parkinson's disease, or ALS or HD, we all have one or more genetically engineered mouse models that are available for study. For example, in the case of Alzheimer's disease, we now already have 3 different mouse models, FAD2T, FAD3T, and FAD4T. Different numbers represent the different counts of mutations carried by each model. And for our ALS mouse models, we constructed them based on the different causative genes. For example, we introduced the human mutant genes like the SOD1 gene with the mutations of G193A and all the TLDBP gene with the mutations A315T into the mice to construct the disease mouse model. And in addition to the gene editor mouse models, we also offer the virus, drug, or the surgery-induced disease mouse model. For example, the pain disease mouse model and the depression mouse models we just mentioned are constructed through the induction method. And here, these mouse models marked in orange means that they are already available, while the others are still under development. Also, we use the clinical indicators to validate how closely the mouse models matches the clinical patients. For example, in the vincristine-induced pain model, we perform the skin biopsies on the mice, just as is done in patients. And this method reliably demonstrates a decrease in the NF expression in the model mice, confirming the parallel to the human disease. Okay, let's now begin our main discussions on amyotrophic lateral sclerosis disease and the associated mouse models. ALS was first described in the 19th century and is also known as low-grade disease. Over time, it has been extensively studied. However, many aspects remain mysterious. ALS is a progressive neurodegenerative disease that affects both upper motor neuron and the lower motor neurons. It leads to the muscle atrophy, muscle weakness, and eventually paralysis, significantly impacting the patient's quality of life. The clinical features of ALS are associated with the damage to the voluntary muscles, leading to the progressive limb weakness, speech and swallowing difficulties, and some patients will with the respiratory failure, also along with the muscle atrophy and hypotonia. These motor dysfunctions result from the combined degeneration of upper motor neurons in the brain motor cortex and some lower motor neurons in the brainstem and the spinal cord. Although the primary symptoms of ALS are related to the motor dysfunctions, up to 50% of patients develop the congenitive and behavior impairments together during the disease course, and also with about 30% progressing to the behavior variant frontotemporal dementia. Given its severe consequence, so understanding the ALS is crucial for developing effective treatment and providing support to patients and their families. Early recongenation and intervention can also improve the outcomes. The global incidence of ALS is about 1.5 to 2.5 per 100,000 persons per year, and a prevalence of 5 to 6 cases per 100,000 people. And it primarily affects individuals aged from 50 to 70 years old and with a slight male predominance. Most cases are sporadic, while only 5 to 10% are linked to the genetic mutations. Some environmental risk factors, including smoking or plastic exposure, whereas the high BMI or the diabetes may have the protective effect. And the median survival is typically 2 to 5 years, with worse prognosis in older patients. and the geographic variations and the gene-environmental interactions remains the key research focus, and there are currently no effective preventive or curative treatment. Evidence from both clinical and basic research shows that the ALS has complex causes, with genetic factors playing a significant and viral role. And up to 10% of ALS patients have at least one other affected family members, and I defined it as the familial ALS, which is almost always dominantly inherited, and the remaining 19 to 95% are sporadic cases with no family history. And with advancement in the technology, the molecular genetic techniques are increasingly being applied to the ALS research. Although over 50 genes potentially associated with disease causation or modification have been identified, the disease-causing mutations in SOD1, C9 ORF72, and FARS and TRDBP are the most common genes, while mutations in other genes are relatively rare. While there is still no cure for ALS, several drugs have been approved that can help to slow the disease progression and manage the ALS symptoms. So today I will briefly introduce three key pharmacological agents, riluzole, edaravone, and tofsen. First one is riluzole, which is approved in 1995. It was the first drug to show a survival benefit in ALS. Its primary mechanism is believed to be the reduction of glutamate-induced excitotoxicity in the nervous system. Essentially, it helps to decrease the damage caused by the overactive nerve signaling. And clinically, riluzole have been shown to extend the survival by several months and delay the need for the tracheostomy. Next, we have edaravone, which received approval in 2017. This drug tackles the disease from a different angle. It's a potent antioxidant that scavenges free radicals. By reducing the oxidative stress, which is a significant contributor to the motor neuron damage in ALS patients, edaravone helps to slow the decline in daily functioning. And it's important to note that its clinical trials showed that most significant benefit for the specific subset of patients with early onset disease. And finally, the most recent advancement comes with tofsen. This represents a groundbreaking shift in our approach because it's a gene-targeted therapy. Tofsen is an antisense nucleotide designed specifically for the patients with ALS linked to mutations in the SOD1 gene. It works by directly targeting the RNA instruction for the 40 SOD1 protein, reducing its production. Why not a cure? Clinical trials have shown that tofsen can significantly slow disease progression, and most importantly, it can reduce the levels of the neurofilament light chain protein, which is the key biomarker of neural damage in these SOD1 specific patients. So in summary, these three therapeutic approaches has evolved from broad neuroprotection with the riluzole and to targeting the oxidative stress with edaravone, and now to the precision medicine with tofsen for genetically defined populations. Beyond the currently approved these medications, the therapeutic pipeline for ALS is highly active and promising. A significant number of investigation drugs are now in various stages of clinical or preclinical testing. These new candidates are particularly exciting because they are appearing novel strategies, moving beyond the traditional approaches to tackle the entirely new mechanisms and new pathways implicated in ALS. For instance, we are seeing advanced research in several key areas. First, targeting the neuroinflammation. There are compounds designed to modulate the immune system within the central nervous system. And the goal here may be to curb the damage inflammatory response driven by the because of the dysfunctional glial cells, which are known to accelerate the motor neuron injury. And second, the development of multi-target therapies. Unlike the drugs with a single mechanism, these innovative approaches aim to simultaneously address multiple pathological drivers of ALS, such as combining antioxidant, anti-excitotoxic, and the neuroprotective effects in a single therapeutic agents. Also, it's not included the MS 0035. And third, exploring advanced modalities like cell therapies. This involves the transplantation of some specialized cells to protect the surviving neurons, reducing inflammation, or even provide support to re-establish neural circuits. And this progress highlights a promising move towards more personalized and mechanism-driven treatment for the ALS patient. And most models of ALS have played an indispensable role in the discovery and development of these therapeutics. So to further empower and accelerate the ALS drug discovery, GemPharmatech has engineered a comprehensive suite of preclinical ALS mouse models. This suite includes the models targeting the key genetic drivers of this disease. The first one is the B6 HSOD1 G93A HSOD1 mouse model. And this model was generated using the transgenic strategy, which co-expressed the human wild-type SOD1 gene and the human SOD1 gene with G93A mutation. After generating this mouse model, we first validate it at the protein level. The Western blot results suggested that human SOD1 protein and the misfolded human SOD1 protein can be detected in different brain regions like the hippocampus and cortex and the cerebellum, and also in the muscles and hearts of the SOD1 mouse model. For further validation of this model, we conducted a series of tests on physiological and pathological metrics. We first focused on the body weight and the lifespan of these mice. A progressive reduction in body weight was observed in male mice, starting from 5.5 months of age, with an initial 2.5% body weight loss, and a statistically significant difference compared to the control group was established by 6.5 months of age. And in contrast, the onset of significant weight loss in female mice was delayed, occurring at around 7 months of age. And regarding the survival curve, the median survival age was around 8 months of age for both male and female mouse model. To assess the motor deficit in ALS patients, we employed two primary motor function behavior tests in the mice. The first one is the grip stress test, which could measure the limb muscle stress. And the results show that model mice exhibited a consistent decline in limb strength starting from 3 months of age. And the second is the root test, which evaluates the motor coordination and balance of the mice. The results demonstrated that the model mice began to show a stable reduction in motor coordination from 5 months of age. And this was specifically evidenced by the significantly short latency to fall from the root compared to the control group's mice. And together, these two findings from the behavior test indicated that the model mice successfully recapitulate the progressive motor impairments characteristics of the ALS patient. We also conducted a pathological examination of the muscle tissue in these mice. The SOD1 male mice exhibited the significant muscle atrophy beginning from 3 months of age, manifested as the muscle fiber necrosis, muscle cell atrophy, and the inflammatory cells infiltration. Loss of motor neurons in the spinal cord was labeled by CHAT as a biomarker, and in the spinal cord was observed to occur at about 7 months of age. To investigate the neuroinflammatory response, we performed the immunohistochemistry staining for GFAP and the IBA1 to label the astrocytes and the microglia cells in the spinal cord, respectively. Quantitative analysis revealed that a significant increase in the number of both cell types in 7 months of old model mice compared to the controls. And this upregulation indicates the glysis of glial cells and provides the clear evidence of the ongoing neuroinflammations in the central nervous system of these model mice. And finally, we collected the spinal cord tissue from the mice for the transcriptomic analysis. The gene ontology analysis of the biological function of the differentially expressed genes also reveals that the immune response are highly activated in the SOD1 mouse models after the disease onset. And these spinal cord tissues are collected from the 6-month-old mouse models. Here the slide shows that the key difference between the GemPharmatech's SOD1 mouse model and the other models from the market. And a notable distinction is the later onset of the disease phenotypes in our model. Pathological changes typically begin to manifest at about 5.5 months of age, followed by a disease progression period of about 2.5 months. And this delayed onset and extended progression timeline consequently provide a significantly longer therapeutic window for efficacy evolution compared to the other mouse models in the market. And the second is ALS mouse model is TDP43 A315T mouse model. And this strain was established by co-expressing the Y-type human TRDVP cDNA and the human TRDVP with A315T mutations. Similarly, the expression of the human TDP43 protein was detected only in the model mice. And in contrast to the SOD1 mouse model, the TDP43 mouse model exhibits a robust expression of human TDP43 protein. And this is highly relevant because in the human ALS patient, key pathological features of TDP43 is its mislocalization from the nucleus to the cytoplasm. In healthy neurons, the TDP43 is predominantly located within the nucleus. However, in the disease state, the TDP-43 protein, particularly its phosphorylated form, mislocalized to the cytoplasm. There is eventually forms toxic aggregates, and which are a hallmark of the disease, and ultimately leads to the neural cell death. And here are the representative images of TDP-43 inclusions in the cortex and the spinal cord from the 3 to 9 months old YTAP and TDP-43 model mice. And a significantly lower body weight increase compared to the wild-type mice beginning at 3 months of age. But the mice did not lose their own body weight, only show a slight decrease of the body weight changes. The mice exhibited a decline in the grip strength starting at 2 months of age, followed by the impaired motor coordination on the road from 4 months of age. We then collected the gastrocnemius muscle from the mice and performed the weight measurement. The diameter and the weight mass of the calf muscle decreased progressively beginning at 2 months of age. Also, TDP-43 mouse models exhibit the significant skeletal muscle atrophy from 3 months of age, manifested as the myotrophy and the nucleosis. And the neural damage biomarker neurofilament light chain (NFL) was first detected at elevated levels in model mice as early as 8 weeks of age. And this serum NFL levels showed a sustained increase, continued to rise throughout the observation period until at least 30 weeks of age. And compared to the mouse models in the other TDP-43 mouse models in the market, the GemPharmatech TDP-43 mouse model demonstrates a significantly earlier age of disease onset and a notably extended overall survival period. And furthermore, we have not observed any mortalities in our model from complications like the intestinal obstruction, which is something that often happens in other TDP-43 models. And for the TDP-43 mouse model, we also have clients who have conducted the head-to-head comparisons using our model alongside with the model from Jackson Lab. And based on the grip stress test results, our mouse models exhibited a constant and significant decline in grip stress compared to the wild-type controls, starting as early as 2 months of age. And the defect was stable and persisted throughout the observation period until at least 10 months of age. And based on the gait scoring analysis, GemPharmatech's mouse models exhibit an earlier onset of gait abnormalities, which are detectable starting from 2 months of age. And in the road test, both models initially exhibited a decline in motor coordination at a similar age, around 4 months of age. However, GemPharmatech's TDP-43 mouse model demonstrated a more stable and persistent phenotypic progression, with the motor defects consistently observed up to 10 months of age. And the plasma level of NFL was elevated in both models, while the GFAP level showed no significant change. And in conclusion, both SOD1 mouse model and the TDP43 mouse models recapitulate the core pathological features of the ALS disease, including the motor neuron loss, muscle atrophy, and associated motor defects. And the key difference lies in the TDP43 mouse model, which additionally exhibits the hallmarks TDP43 protein pathology, such as mislocalization and the aggregation, which can be observed in the majority of ALS patients. And furthermore, the TDP43 mouse model demonstrates an earlier disease onset, and its progression allows for extended experimental observation as it doesn't feature a rapid survival endpoint. And as mentioned earlier, in addition to the mouse models, GemPharmatech can also provide the preclinical efficacy service. And all the data we have presented here are conducted on our own preclinical efficacy platform. And here is an overview of the service that we can offer. We can provide a series of preclinical drug efficacy evaluation service or the phenotype screening service of some new mouse models based on our preclinical platform, including the drug administration, modeling, behavior analysis, in vivo biochemical detection, sample collection, and in vitro pathological analysis. And throughout the entire workflow, the first step is the drug administration. In terms of the drug delivery technology, we not only have common injection methods such as IP injection, IV injection, SC injection, but we can also provide some delivery methods specifically for the central nervous system, such as the intracranial injection or the intrathecal injection. This method can help the drugs cross the blood-brain barrier. And based on our experience and some literature, we can provide our clients with a maximum dosage range from the different administration methods. And in terms of the behavior test, if you are interested in the amyotrophic lateral sclerosis or Parkinson's disease, this motor disorder, we have a series of motor functioning testing instruments for you to choose from, such as grip stress, rotarod, treadmill, and get analyzed. If you are more concerned about the congenitive disorders like the Alzheimer's disease, we have classic the Morris-Watt maze, Y-maze, and novel object recongenation, and nesting, and so on. And in addition, we also have the Von-Free test and the hot plate or cold plate test for the pen testing. And also the tail suspension, forced swimming test, and the sugar water preference for the detection of mental disorder, such as depression and the anxiety. And taking the Morris-Watt maze as an example, it's mainly used to evaluate the spatial learning and the memory ability of mice. And the following indicators are used to jointly determine whether the mice have congenitive impairment. For example, our AD mouse model F8040, compared with the Y-type mouse model, Y-type mice, F8040 mice showed natural learning and memory abnormality indicated by the less crossing number and the less time spent in the platform area, and also the target coordinate in the Morris-Watt maze. For in vivo biochemical detection, we can provide the in vivo imaging platform to detecting the drug distribution, and the DEXA to detect the bone mineral density and the body composition. Also, the metabolic cages are used to monitor the physiology status of the mice after drug administration. Additionally, we can collect virus tissues after experiment, such as the brain regions samples, the spinal cord, and the CSF that commonly used in the neurological study. For in vitro pathological analysis, we can do both histological analysis and the molecular analysis. For histological analysis, we can provide chemogenic staining, immunohistochemistry, and the pathological evolution. And for molecular analysis, we can do the gene and the protein expression analysis by qPCR, Western blot, or ELISA. And also we can do the blood analysis. Here is an example of the immunohistochemistry showing the beta deposition in the AD mouse model. And also the immunofluorescence staining results exhibited the glysis of the astrocytes in hippocampus in the FAD40, the AD mouse model. And that's all the introduction of our ALS mouse models and our platform. And in summary, the GemPharmatech can not only offer ways of models resource, but also serves as a reliable partner for your preclinical efficacy study. If you are interested in any of other models or our platform, please feel free to email us to obtain the detailed data. That's all what I want to share with you today, and thank you for your patience.
Thank you very much, Dr. Lin, for the very insightful and comprehensive presentation. We will now move into our Q&A session. If you have any questions, please feel free to type them in now into the Q&A box, and we'll address them as they come in. So we have already a couple of questions coming in right now. So for the first question we have is, how do the onset and progression of motor deficits in the B6 SOD1 mouse compare with those in the TDP43 mice? And how should researchers choose between these models depending on their therapeutic target?
Thank you for your questions. And for these two different models, the first one is the SOD1 model. This model exhibits later but more rapid and severe progression. And the motor deficits like the grip stress decline typically begin around 2 to 3 months of age, and with significant motor coordination deficits on the road appearing at 4 to 5 months of age. And this disease progression is aggressive and leading to a clear survival endpoint, so make it ideal for the studying neurodegeneration and survival. And for TDP43 mouse model, this model demonstrates an earlier onset, but a more prolonged and stable progression. Like the grip stress test can be detected as early as 2 months of age. And the phenotype is also progressive and stable, allowing for the observation of the deficits from 4 months up to at least 10 months of age, without a rapid survival endpoint. So its key feature is robust TDP43 protein pathology, its mislocalization or its aggregation. So if our clients are interested in the SOD1 specific therapies, like the gene therapies we just mentioned, or the survival endpoint studies, SOD1 mice will be the better choice. And if you are focused on the TDP43 pathology or some long-term efficacy studies, the TDP43 mouse model will be more suitable. And if you are interested just in the neuroinflammation or muscle atrophy, both of these two models can be used for your study.
Oh, wow. Thank you very much, Dr. Lin, for that very detailed description. And I think really definitely it highlights the fact that it really depends on the type therapy that you're working on and the length of study. So thank you very much again. So the second question we have is, at what stage of disease progression in these models, such as early muscle atrophy, motor decline, or neuronal loss, would you recommend initiating therapeutic intervention studies to best reflect clinical treatment windows?
To best mirror the clinical scenario, where treatment begins often or shortly after the symptom onset, we recommend the intervention strategy based on the earliest detectable biomarkers and functional deficits. For example, for the SOD1 mouse model, we recommend the initiating the drug administration from 5 to 5.5 months of age, when the mice begin to exhibit the declines in body weight and the motor functions. And for the TDP-43 mouse model, we will suggest starting treatment from 2 to 3 months of age, when the mice initially show the changes in the pathological phenotypes and also a reduction in the motor ability.
Yeah, thank you very much for that. I think this will definitely inform our listeners when to best utilize these models to be able to, get the
Efficacy.
best efficacy data from their therapeutic agent. So
Yes.
yeah, appreciate that. Absolutely. Thank you very much. The third question we have coming in is, given the heterogeneity of ALS patients, can these models help inform patient stratification in clinical trials? For example, distinguishing therapies for SOD1 mutations carriers versus sporadic ALS.
Yes, these two models all are powerful tools for developing the precision medicine strategies for ALS. The SOD1 mouse model is definitely a preclinical tool for the therapies targeting the SOD1 mediated the ALS. And the TDP-43 mouse models is critical for the therapies aimed at TDP-43 protein pathology. Since the TDP-43 pathology is a hallmark of nearly all the sporadic ALS and many genetic forms, like the C9orf72, these mouse models. So efficacy in these models could suggest a therapy have broad application beyond a single genetic mutation. It can help to identify the treatment for the larger sporadic ALS population.
Okay, no, thank you very again. I'd like to take this chance again to, if you have any questions, again, feel free to enter into the Q&A box. We have one other question. This question comes in and it says, how well do the phenotypic readouts in these models, such as motor decline, weight loss, or protein aggregation, align with clinical biomarkers currently being used or explored in ALS trials? Very
Yeah,
interesting question.
actually, the alignment is very strong. We use the phenotypic readouts, particularly the motor functions and the NFL expression in the plasma, show the expressional translation to the clinical patients. And the progressive motor depth in the mice directly mirrors the functional decline measured by the behavior analyzed in the patients, like the ALS, FRSR, and the rise in the plasma NFL in models could predict and correlates with disease progression, exactly as it does in the patients. And its rapid response to the effective treatment in models, like we just mentioned that the toxin therapy in the SOD1 mice could directly precede its use as a pharmacodynamic biomarker in the human trial. So basically, these models could help us reduce the risk clinical trials. If a drug can lower the NFL levels in our mouse model, it will be a strong sign that it maybe or will probably work in the humans too. And it also give our clients a lot more confidence before they start the expensive human studies.
Oh, okay, thank you very much again for that. It's a very insightful response. So I don't see any other questions coming in from our Q&A. So on behalf of GemPharmatech, I would like to thank you all for joining today's session and for all your thoughtful questions. And again, thank you again to Dr. Lin for her very insightful presentation. We hope this webinar has provided valuable insights into how ALS mouse models can help advance our understanding of the disease and help accelerate therapeutic development of therapies. A recording of this session will be shared with all registrants, and you can also find more information about GemPharmatech's ALS models and preclinical solutions on our website at GemPharmatech.com. I would like to thank you again for joining us today, and we look forward to staying connected and supporting your research. Have a wonderful day.
Thank you. Bye.
Bye.